Welcome back to Monday Biology Day. Right now, I'm sitting in the jungle. Not really, but I brought some of my plants along because guess what today's topic is? We're talking about photosynthesis, as you know. All you Unit 3 biology students out there for VCE, but also any biology students across Australia studying photosynthesis. This is relevant for you guys. This is one of my favourite beauties. I wonder if she could sit on my shoulder while I talk. Photosynthesis. All right, guys. So in biology, we need to understand the idea of living things and how cells work, multiple processes working at the same time to allow for living things. We need to know where we get our energy from. That's it. And you guys in your course are specifically talking about two of the biochemical pathways that are most fundamental to our existence as eukaryotes, and that is photosynthesis, creating glucose and carbohydrates for structural components and also for energy. And so that's your first sort of look at these biochemical pathways, and then looking at cellular respiration, and that's the breakdown of the glucose that was made in photosynthesis. So these two reactions have a very close relationship, and they are both just incredibly mind-blowingly fundamental to our existence as eukaryotes, as multicellular organisms. But we do owe our existence to photosynthesis, basically producing that oxygen and that glucose for all the things that we need to survive. So thank you to the chaos that is our living world. And today I'm just going to talk briefly about photosynthesis as a reaction and where it occurs, and break down the inputs to the reaction and the outputs, because VCAA, they want you to understand the process, but for the purpose of knowing what needs to go in and what needs to come out, what the inputs and outputs. This year they're saying, well, the chemical reaction side of things, well, not this year, this study design, chemical reaction side of things is not required. But for me as a teacher, as a learner, I find, you can see my door there, I find if I don't understand the process, I'm not going to remember random terms like NADPH and ADP and electron acceptor and loaded coenzymes. I'm not going to hold on to that information if I don't understand where they fit in and the story of their role in stuff. So today I'm going to break that down for you. I'm just going to share my screen. We like to share our screen on these lives because when you're teaching biology and chemistry and stuff, it helps to have visuals, as you know, and some of these visuals are quite similar to ones you've seen. I'll just share that screen. There it is. A couple of slides just to help us visualize. Things are in the way. Slideshow. All right. Last week, I spoke to you guys about, but you had no sound if you watched it later, but I spoke to you guys about enzymes and their role in biochemical pathways. And now that was sort of an introduction to think about the fact that chemical reactions are happening throughout your cells. Thousands of reactions are happening at any one time. That's known as your cell metabolism. So all the reactions, all the biochemical pathways occurring in a cell. It's very it's very complex, you know. And so VCAA are telling us, all right, let's break down two of the fundamental reactions that are really important to survival and to life as we know it. And let's look at those reactions, a little bit about what goes in and therefore what comes out, and the fact that it's multi-processed, like multi-stepped, which is the idea of a biochemical pathway. It's not just a chemical reaction, it is a pathway of multiple sort of steps to get to your end product. So I want to talk about photosynthesis as the producer of oxygen and glucose and water, And then next you'll sort of talk about cellular respiration, which is the breakdown of the glucose to make usable energy. All right, so I'm going to stop sort of rambling about VCAR. Plants, though, oh my gosh, they're amazing. They're just amazing. You can see the size of these leaves here and the positioning of the leaves, right? So these leaves are quite large. And, you know, plants come in all shapes and sizes, as you know. This is not a plant list, but it kind of is because we're talking about photosynthesis. This is one of my favourite pots. It was given to me by a colleague. So we are looking at parts of the plant that allow for photosynthesis to happen. Where does a plant get the nutrients and the materials it needs to do photosynthesis? And the next video will be also different types of plants and what can affect their rate of photosynthesis, because what we're talking about here is like there's plants, there's small shrubs, there's tropical, beautiful flowering plants, there's, you know, 50-meter tall trees. There's a range of plant life out there and they survive in various different conditions. So photosynthesis today is the general idea of it in terms of C3 plants, which is like about 85% of plants on the planet. And then you will look at a couple of other different examples called C4 plants and CAM plants a bit later. All right, so quick general reminder about a plant. If you look at our diagram too, you've got sunlight on the left, comes in and interacts with the leaves and the green parts of the plant. Obviously, sunlight doesn't get below the soil, so the roots are not drawn as green. The roots are often like a colourless kind of whitish kind of colour. The roots are not exposed to sunlight, so they're not using that. You've got carbon dioxide in the atmosphere in grey here, in the atmosphere that enters the plant. You've also got water that enters the plant. So these things the plant takes in from the environment. The plant releases oxygen out back into the atmosphere, so gas exchange there through the leaves. It doesn't sort of excrete glucose, but it produces glucose, and that travels all around the plant because glucose is needed as an energy source down in the roots, in the trunk, the tips, in all the growing areas, in like everywhere. All cells need glucose, and also water is also released. So just a reminder about the fact that, you know, leaf structures, so when it's the way that a plant gets its gases that it needs, so it will take in carbon dioxide and release oxygen, there's gas movement in and out of plants via their leaves, underneath their leaves called the stomata, so little pores, like the pores in your skin. You've got little pores under there that allow movement in and out of oxygen and of carbon dioxide and gas, so that we call that gas exchange. But when it's raining and you've got leaves covered in water, those leaves are not taking in water. They're not absorbing the water as part of their requirement of water. Water is drawn up through the roots and moves in that upward direction and reaches all of the cells through a process called transpiration. That's something you sort of study in year 11. But it's just a reminder that water is exiting the leaves. I can't say leaves the leaves because it's too hard to say. So water exits the leaves. Water does not go in the leaves, so it's one direction. So if a plant wanted to reduce water loss on a really, really hot day and or in really arid environments, we could close those pores and that would stop the water evaporating out. That is key to some of the adaptations depending on your environment. So that's coming as well next week. All right, I'm going to put him down. How cool is he? Thank you for my teacher friend who had me for KK. So just quickly, in terms of where this amazing reaction is happening, this reaction is happening in the chloroplasts in plant cells. There are many chloroplasts in one plant cell. So there's lots and lots of chloroplasts. And you need to know the general structure. So you've got this double membraned little compartment, little organelle that has the inner membrane, the outer membrane there, and it looks like a bit of a bean bean shape. And if you kind of take a cross section in there of that bean, take the top off, you can see that you've got all of these little, they look like discs or coins, little stacks of thylakoid. So basically, thylakoid is a membrane. It's a type of membrane that is made of little phospholipids. So just like your little men with the legs, just like your cell membranes and things. But in fact, it has different components as well. So it's not like the plasma membrane because it has some extra different components that don't exist in a plasma membrane. For example, chlorophyll. So these discs in the chloroplast, you've got to imagine them like a 3D structure, and then if you cut into them, that sort of shows you there's sort of like a hollow space in the middle, right? So these are all made of this continuous membrane called thylakoid membrane, and they form these discs, these stacks. One stack is 1 granum, and many stacks are the grana. So this is where the grana and the thylakoid membrane, that is where you're seeing the first part of the reaction, first part of photosynthesis, which is called the light-dependent stage of photosynthesis. It's happening in those discs. And then the space all around those discs is called the stroma, with an R, stroma. That is where your second part of the reaction is happening. Your light independent or Calvin cycle is happening there. So you've just got to have an idea of where these different stages happen. And again, not like the in-depth biochemical pathway of each thing, because there's many steps involved, but you have to have the general idea of what goes in, what's an input, and what's an output. Also, so chloroplasts, we always see them as green. Why are these amazing plants green? This one actually looks like it's so healthy it looks artificial, but this was a gift as well from a little nephew. So why are they green? Anything that has colour basically is sort of reflecting wavelengths of light back to your eyes at a particular wavelength that resonates with the brain, and we identify it as a colour because we're taught over the years, hey, this colour is green. Look at this red jumper. Isn't this a beautiful purple something? So you learn the colours based on what you're seeing, but colour is basically just the light being reflected back into your eyes at a certain wavelength. That's a whole nother story, and it's so cool. So if you don't really know much about that yet, go find someone like Brian Cox or someone like that. Really cool thing to look into about the visible light spectrum and how our eyes work in that way. So the point of that is, why are plants green? Why are chloroplasts always drawn as a green? It's because chlorophyll is a pigment there that is absorbing certain parts of the light and reflecting other parts of the light. So it's reflecting that green light, that yellow light. That's why the plants there's a lot of different greens. But there's variation in types of chlorophyll. So it's like the ratio of like chlorophyll A and chlorophyll B is that combination, and therefore the light reflection that gives you the different shades of green. And also the depth of the green is dependent on how much light is available. And often, like if you go through a rainforest, often you might see darker greens towards the bottom because they've got more chloroplasts to try and capture that limited light that's being shaded by all those tall trees, in the upper canopies of the rainforest. So just pay attention to nature. She's talking to you and telling you all the time. Cool. So that's the structure of where photosynthesis is happening, the chloroplast. I'm talking quite a bit. I'm going to jump to, you would have seen this diagram a lot, guys. It's sort of reimagined for us, but it's a good summary diagram of photosynthesis. So there's two parts to the reaction. The first part is depending on light. It needs light to work. So it's called the light-dependent stage. That is necessary to produce some products which are then used in the Calvin cycle or the light-independent stage. So the second reaction won't happen without that first one, without the outputs of the first one. But the second reaction does not use light itself. Okay, so it does rely on that first reaction happening, but it's not light dependent itself. And you can see it's all happening within the chloroplast, and there's this shuffle of molecules here. You've got NADPH being produced in the thylakoid in a light dependent reaction, which is then, you can see the arrow there, it's going to be used in that second reaction. You've got ATP energy going from that first one being produced and then being used in the Calvin cycle. And you can see, follow that around, you can see that ATP, adenosine triphosphate, dropped off some energy, and in doing that formed adenosine diphosphate and inorganic phosphate. So we just break off that third phosphate. That energy that's released is used to drive the cycle, and then ADP and PI will go back to the light dependent stage. It'll just be floating around the area. It'll be loaded up again, and the cycle continues, right? So the inputs, basically, for this slide, I just want to say that the products of the light dependent stage, some of them are needed for the second stage, others are released as waste, for example, oxygen. Isn't that funny that oxygen is seen as a waste product for photosynthesis? But it's the elixir of life for us. So all I'm going to talk about today for the rest of the time is just break down the light dependent reaction and then the light independent reaction. Again, for your course, you need to remember the ingredients, what the inputs for photosynthesis and the outputs. But for me to remember those things, I need to sort of have it as a story in my mind. So I'm going to tell you the story of photosynthesis. Okay. Just going to check that I can see any comments. Not really. Cool. Okay, guys. So first reaction. You will see this diagram in various forms. Have a look to the very left. Don't be intimidated by all of what's going on. Have a look to the left. That's another sort of representation of a chloroplast, and you can see those stacks of thylakoid membrane there producing the grana. And here's our little box saying, let's zoom in to just that little part of the thylakoid membrane. So here is the thylakoid membrane, a little section of it, and you can imagine this is on repeat. There's thylakoid. This is, this part of the membrane is called the electron transport chain, which transports electrons. And so you can see throughout the thylakoid, you've got electron transport chain, you've got all this sort of parts of the membrane repeated. So photosynthesis is happening like a lot in just one chloroplast. And imagine how many chloroplasts are in this plant. Each plant cell that we can't see, each cell has multiple chloroplasts, and each chloroplast has multiple grana. And think about how you get like the tree out in your backyard that's 10 years old. How tall is it? How many leaves does it have? This reaction is happening all over the planet. It's just incredible. So zooming in just to a picture of the membrane, you can see the orange little phospholipids. You got your phospholipid bilayer, right? So that's just like your plasma membrane. But we have some different proteins and different pigments in here that mean that this is called thylakoid membrane instead of plasma membrane, okay? The two green bits are chlorophyll, those pigments embedded within the membrane, and then you've got these other pink proteins scattered throughout. So I'm just going to tell you this story. You do need to know the inputs and outputs. VCAR doesn't need you to remember the name of the types of chlorophyll or anything like that, but you need to know the story. So chlorophyll here is a pigment that absorbs red and blue light wavelengths of light from the sun, and it reflects or bounces off, reflects back green and those sort of yellows. It doesn't use that light very efficiently. It doesn't absorb it and use it to energize, right? So That's why we're seeing them as sort of green. That's definitely something you'll probably look at in class in terms of factors that affect it. So light, the colour of light, the wavelengths of light affect this reaction. So you have your chlorophyll here, photosystem 2 and photosystem 1. So PS, not PlayStation, PS2. You've got light, artificial light, sunlight, whatever the type of light, doesn't matter. Wavelengths of light are hitting that membrane and hitting that chlorophyll and exciting electrons there. Now, if we get our electrons excited, you can see the arrows there, they follow, they pass, the electrons move from pigment to proteins and stuff. They get passed along this chain. It's the electron transport chain, transporting electrons through, and that has consequences. And if you were to, as a molecule, just as a compound, just keep losing electrons, you would collapse and you wouldn't basically like function anymore. You wouldn't be viable. So it's not like chlorophyll is just losing electrons and everything's fine. There needs to be a source of electrons to go through this chain. And so that source of electrons is the water. So we've got an enzyme there attached to chlorophyll. The enzyme helps split water. And what's in water is hydrogen and oxygen, all right? H2, 2 hydrogens, and oxygen. So we have water that's split to form oxygen and hydrogen, and that process releases electrons that are taken up into the chlorophyll, and those electrons are excited by light, and they get the energy to pass through the chain. So water is split. I'm using my language very consciously, guys, because you need to be using these sort of these terms as I go through. Water is split to create oxygen and hydrogen. Hydrogen stays and builds up the concentration gradient here, the concentration of hydrogen. Oxygen is released as waste. It's not that it's necessarily released, because the plant cells use oxygen themselves as well, which you'll find out very soon, for like in the mitochondria. But it's released as waste, we say, because it's not then used in the next reaction. It's not like part of the products, right? So the product of glucose. Cool. Water is split, provides electrons to the chlorophyll. Light excites electrons, and they move through the chain. You'll notice that we have a proton pump here. Protons are positive. There we go. We've got our hydrogen, our H plus ion, which is a proton. Positive ion there goes through the proton pump. And proton pumps, so remember that from maybe if you did year 11, the membrane has its own properties, hydrophilic and hydrophobic properties, etc., with lipids. And so hydrogen ions, if they were to try and cross the membrane, they would just be rejected basically and not pass through because they are positively charged. So hydrogen ions can cross the membrane into the lumen there via a proton pump, forcing them through, which is every time this, like an electron goes through the chain, it will power the proton pump to push through hydrogen. And I'll come back to the buildup of hydrogen in a minute. But we're splitting water and providing hydrogen, and our proton pumps are providing hydrogen here to build up this gradient, okay? Back to the electron. So the electron's excited, goes through the system, loses energy as it goes, obviously, because like you were running a race, you'd also get a bit more tired towards the end. It hits photosystem 2, gets re-energized, and will pass along the chain. And basically, the electrons need to go somewhere. If the electrons were going through the electron transport chain and then just sticking around at the end, they would start to bank up like a traffic jam. So the electrons need to be removed or accepted by other molecules for the system to continue. So we have NADP plus here. We have this electron acceptor, this coenzyme that is there to pick up 2 electrons. And in doing that, because itself is positively charged, if it picks up 1 electron, it becomes neutral, but it picks up 2 electrons, so therefore it becomes negatively charged, 1 negative charge. And what's all in the area are these positive hydrogen ions. And so when NADP plus picks up the 2 electrons and becomes NADP negative, it attracts a positive, which is hydrogen, and becomes this beautiful neutral NADPH molecule. A loaded electron acceptor or a loaded electron carrier, which is going to act as a coenzyme, which is going to work with enzymes in the next reaction. So one of the inputs for your light-dependent reaction is NADP plus. This is where it's needed. It accepts electrons from the electron transport chain and becomes NADPH. That's used for the next reaction, which is a light-independent reaction. Okay, so far, guys, my story has already told you about the inputs and outputs of 2 different areas. So you've got water as an input. Why is that needed? Well, water is split to form oxygen, hydrogen, and provide electrons to drive this process. So there's your input of water and output of oxygen, and you've got hydrogen buildup. We've got our electrons being accepted or taken out of the system by NADP plus, and in doing that, it attracts hydrogen right next to it, probably. Hey, hydrogen, come to me, and because it's positive attracts negative, you get NADPH, and that's used in the next reaction. The last part of the story, or the last part of your list of inputs and outputs, is really talking about ATP, right? So that's where the hydrogen gradient comes in. If you remember, like last year, talking about, or in unit 1, talking about a concentration gradient across the membrane. And so if there's a difference in concentration, usually molecules will move from a region of high concentration to a region of low concentration. So in diffusion, it's the movement of particles from high to low. In osmosis, it's the movement of water from a high water concentration to a low water concentration. So that principle applies here, right? You've got this buildup of hydrogen in the lumen, in that space, and you can see, like, so this is like the inside of the disc, whereas the stroma is the space outside of those grana, outside that thylakoid. So you've got hydrogen building up because water is splitting, and because the proton pump's forcing hydrogen every time an electron goes through. So you get all this hydrogen, and ions, atoms, molecules, they move, they constantly move towards equilibrium. So they spread out, and hydrogen ions cannot cross the membrane directly. So how will hydrogen then move through the space towards equilibrium, towards an equal concentration on both sides? The only way out for hydrogen is through this big protein here called ATP synthase. So ATP synthase is an enzyme. It ends in A's, so that's our key identifier. And if you remember back in the other unit of like DNA polymerase and RNA polymerase, those enzymes make molecules of DNA and RNA. So this enzyme here in this beautiful pink color is ATP synthase. It's synthesizing ATP. So hydrogen can pass through this protein. Remember, an enzyme is a protein. Can pass through the protein, and as it does pass through, it powers the enzyme to fix inorganic phosphate, PI, inorganic phosphate, to adenosine diphosphate, ADP. So the two come together to form adenosine triphosphate, ATP. And ATP is used in the next reaction. So again, hydrogen ion buildup in concentration. Hydrogen then goes through ATP synthase, which powers the creation of ATP, which is needed in the second reaction. So again, VCAR doesn't want you necessarily to tell that whole story, but if you don't sort of have a story in your mind of like what goes in and what comes out and why, then I don't think you're going to find it easy just to remember letters and numbers and NADPH. Like, what does that even mean? What's ATP? So it helps to tell the story. Try and tell someone else the story and see if you can explain where your inputs go and where your outputs are from. That'd be cool. Okay, the last little part is talking about what happens to these products now. And that is the second part of photosynthesis, and that's the light independent stage, which means it doesn't depend on light. So again, this kind of diagrammatical representation of the reaction is very common because it's a really clear way to show that this is a cyclical reaction. Photosynthesis is a multi-step biochemical pathway with many, many steps. And in particular, we look at the Calvin cycle as a cycle, right? So molecules and inputs come in, they undergo a series of reactions, some manipulation there, and then some of those products are turned into our glucose, and some of them continue through the cycle and replace for the next round. So it's called a cycle. You don't need to know all the steps involved because they're not even I'm just I was just picturing, I had a flashback of myself at university having to learn all the steps and going, wow, this is, yeah, and cellular respiration, there's many, many steps in those reactions. So thankfully for us, we need the summary, the idea of what's happening. So we see this as a cycle. You can see the arrows going around. What's going on here is carbon dioxide is in the atmosphere. Where does carbon dioxide enter the plant? Remember my fun friend here? Underneath the leaves, you have all these little pores called stomata, not to be confused with stroma, which is the space in the chloroplast. Gas exchange happens in the underside of the leaves where there's pores, stomata. So carbon dioxide is coming from the atmosphere going into the leaves. Rubisco is an enzyme. You're testing me now. Ribulose by phosphate carboxylase oxygenase. But you guys can call it Rubisco. It's an enzyme that will take carbon dioxide and fix it or attach it to an existing molecule in the chloroplast called RUBP. It will undergo carbon fixation. So you've got a carbon in the carbon dioxide, you've got a 5-carbon molecule. When you bond them together, you get a 6-carbon molecule. And that's called carbon fixation. When you fix, like you might say, I need to fix the TV to the wall, which is like mounting it to the wall, right? You're fixing things together. So that's stage 1 is called carbon fixation, where Rubisco bonds carbon dioxide to RUBP. That then is a 6-carbon molecule. It's stable. It will separate, create 2 3-carbon molecules. Obviously, if you had 6 to start with and it separates in half, you've got now 2 with 3 carbons each. And there's a series of steps that those molecules undergo. We won't go into all the detail. There's quite a few videos out there that if you're interested, but in terms of your course, what you need to sort of focus on is the idea that those molecules here, like you've got your 3 PGA, if you look on the right-hand side diagram. Now, if you're building up a molecule of glucose has 6 carbons. Its formula is C6H12O6, 6-carbon H12 hydrogen, 6 oxygen. And I sort of say to myself, it's CHO, 612-6, CHO, CHO. So glucose, right, is a big molecule that requires even 12 hydrogens, right? It's got to have energy input to create these bonds to build up this nice, beautiful, big glucose molecule. So where does that stuff come from? Well, it comes from your light-dependent stage. So you can see in this main diagram in the middle, Rubisco fixes carbon dioxide together to the RUBP. They undergo a series of modifications, like manipulations, and we call that stage reduction, When you accept or gain electrons, it means you're reduced, which is very much a chemistry term, but the reduction stage is where this molecule accepts electrons from NADPH. So NADPH will drop off those electrons and the hydrogen, and it will turn back into NADP+. That NADP+ will then be around the area. It will then make its way, it will be used again in the light dependent stage to make more NADPH, and that's a cycle. So it's like a shuttle bus almost, bringing electrons and hydrogen to the next part and then offloading them or unloading them, and then going back to reload and coming back and unloading. So in this form, NADP+ is like unloaded electron carrier. To build up a big molecule, right, to put things together, you need energy input to form those bonds, and that's where ATP comes in. So we've got ATP from the last reaction providing energy to build up this big glucose, and when it drops off that energy by the third phosphate, adenosine triphosphate, the third phosphate breaking off, being released, and the energy that's in that bond goes into building up the molecule. And so then the ADP, and we should have a little PI here too, inorganic phosphate, the ADP will then go back to where ATP synthase is, and it will be grabbing another inorganic phosphate. It'll cycle from ATP to ATP, from ATP back to ADP, back to ATP. It's pretty cool. So energy is needed to build up this molecule of glucose. And so remember that your chemical reaction for this is 6 CO2, 6 carbon dioxide. And you've got 6 carbons in your glucose molecule. So it's C6H12O6. But you've also got this RUBP, which is a 5 carbon molecule. So this Calvin cycle continues to cycle through with 6 different carbon dioxide molecules coming in individually. For every 3 carbon dioxide here, we've got our 6 NADP, H, etc. VCAA have said, don't worry so much about numbers and following through, but there are some key numbers that we need to know over, like, for cellular respiration and stuff. But basically, some of that becomes glucose or a carbohydrate for use by the cell, and some of it is used to regenerate or make more of the RUBP, that 5 carbon sugar. So the stages are carbon fixation, where Rubisco grabs carbon dioxide that's come in through the underside of the leaves, through the stomata. Where's my plant? It's very rigid and full of water. Gas exchange happening under here. Then it undergoes a series of reactions with inputs from ATP and NADPH from the previous reaction that you made. Some of that will become glucose, some of it will continue in the cycle and regenerate RUBP, ready for the next cycle of carbon fixation. Cool. So that's the Calvin cycle. That glucose, or those carbohydrates, can then be used for different things in the plant. Either it can store it, because there's going to be lots of it happening. Photosynthesis is happening at a major rate all the time, that there's light, so it can store it as starch. It can use the glucose and make cellulose. Sorry, I'm just checking a comment. It can be used as cellulose, so for structural things in the plant. If you think about it, right, a plant can go from just being this small, right, to just growing and producing leaves and leaves and leaves and leaves and leaves, right? Look how amazing that vine is. Where does it get the material to make all those cells? How does a tree grow to be 100 meters tall? Takes nutrients out of the soil, uses water from the environment, but it's producing its own components in glucose. It's using that then for energy to make other reactions happen. Where does it get the material from? It doesn't eat. Plants don't eat as such, do they? We eat. So it's just amazing to think how intricate plants are, how big they can grow, how extravagantly detailed and colourful and amazing they are. And they're using they're harnessing the sun's energy for a major part of their growth. Very cool. Okay, lastly, before I go, again, inputs and outputs. If you can tell the story, it'll help you remember the inputs and outputs. So the light-dependent stage happening in the thylakoid, water and ADP plus, ADP and inorganic phosphate are inputs of that first electron transport chain phase. Look at what you, is that a typo? No, it's not. And then you go down and you've got oxygen, ATP and ADPH. So that's your general summary table, guys. But I think it'd be cool if you could tell someone your story to help you remember as well. Okay, that's me. I'm going to stop sharing. That's photosynthesis. Next week, we will talk a bit more about the different types of plants and how they might adapt to things like living in an arid desert environment where there's not enough water. So you can't just like lose water through your leaves all the time because that's losing water. So you've got to have other ways of getting the gases in, because if gases are happening, gases are entering, you've got gas exchange happening under the leaves in the pores. Remember I said at the start, water is drawn up from the roots all the way up to then make it to the leaves. If you're shutting off those stomata, those pores, to stop water exiting the leaves, you're also shutting off the entry point for carbon dioxide. So how are you going to do this photosynthesis and how are you going to get the materials you need to grow? That's what we'll talk about next week. Okay, stop sharing. Thanks for listening, guys. Hopefully the sound worked this week. And I'll see you next week, every Monday for Year 12s this term. Love to see you here watching us live. Hope that was helpful. See you next week.